Well, hello, and tonight we're going to be looking at the development of the fertilized egg, and we need to look at this very brief diagram at first. And this diagram is going to just give you a preview of what we're going to be looking at. And so we start with the fertilization of the sperm and the egg. That probably won't come as any surprise to you. And we then get to a zygote, and again, probably familiar to you. And the zygote will start dividing, and as they divide, we start to see many, many, many cells, and that's called cleavage. Now, at this point, we haven't really seen much differentiation, although some cells do have different proteins and different genes turned on. So we're going to start seeing a really major shift in gastrulation, and you see the blastula turning into the gastrula, and that's where we see the development of the digestive system. And then from the digestive system, we start seeing all of the other organs being developed, and then we get to a very, very beginnings of the embryo that maybe some of you recognize. So let's get started, and we're going to get started with fertilization in a little bit more detail. And so let me zoom in on this a little bit so you can see it. And fertilization is going to be starting with our sperm cell binding to this jelly-like coating of the egg. And the jelly-like coating is going to be a combination of the extracellular matrix that we learned about before. And this extracellular matrix is going to be a combination of glycoproteins and water, and that kind of creates this jelly-like substance. And so when the sperm binds to this jelly-like coating, it starts something called the acrosomal reaction. And the acrosomal reaction is so named because of this little part that is labeled orange here, orangish red, and that is called the acrosome. And the acrosome is going to contain enzymes, and these hydrolytic enzymes are going to be released. And you can see that in this picture right here. This diagram is showing things much like in a time lapse. So they've taken different pictures, snapshots, and it's indicating what's happening in a stepwise fashion. So we see these enzymes slowly dissolving away the jelly-like coating here. And in this next stage, we're seeing the cell cytoskeleton, these proteins are extending, and we see proteins binding with this protein on the egg membrane. And this is an important point of recognition. And you might even recall we talked about this earlier in the year with the sea urchin speciation, where one or several mutations that change the shape of the protein, and this change of shape of the protein would then dictate a lack of recognition of the sperm cells to the egg. And once we have that, we have two distinct populations, reproductive isolation. And that's important for all of these species that especially ones that externally fertilize. Now, the other event we start seeing is the cortical reaction or the cortical process. The cortical reaction is going to prevent polyspermy. And this is going to be really important. We do not want more than one sperm fertilizing an egg. That would be a triploid, and animals just really can't handle that. It's just not going to form. It's not going to develop. So we want to block other sperm cells. And so what we start seeing as that sperm penetrates that egg, this is going to set off these cortical granules right here. And these cortical granules, they also contain enzymes, different enzymes, and other molecules that will be released in this space. And as they are released, it's also going to start causing water to flow in, and it's going to make this extra thick layer. And not only that, but it's going to serve to harden this region of the egg, and we will then see all of the other sperm cell kind of being pushed away, and then the sperm cells can't fertilize. And you can see this in this diagram over here. Let me zoom in. and we can examine it in a little bit more detail. And what you're seeing as little white wavy lines in this picture, these are actually sperm cells. And you can see here would be where the sperm cell has fertilized that egg. And how do we know? Well, we know because that's where the cortical reaction has started. And you can actually see this membrane starting, and it wraps around the cell. And as it wraps around the cell, notice all of these sperm cells are being pushed and blocked. And at this point, that egg is fertilized. We now have a zygote. And now we can start looking at the details of the development. Now, the first thing to notice is this time of very rapid cell division. And this very rapid cell division is called cleavage. We see now this cell division is mitosis, because at this point, the cell does not need to have its genome. It's fine. So it's now diploid, and we're going to stay diploid. We're just going to divide. And it's so rapid that it skips both G1 and G2. A little review point, during the cell cycle, we start with G1, we enter into S, which is the stage where DNA duplicates. We go into another gap phase or growth phase, G2, where everything gets prepared normally. And then we go into mitosis, and then it goes right back into G1. Well, in the zygote, these steps are skipped. Now, part of the reason it can do that is this cell is big, and you've got this very large bundle of cytoplasm, and so you don't really need the growth anymore because it's there. But what will happen is you've got this very large cell, and it divides into 2 and then 4, and then you can continue the math progression until you get to about a ball of 100 or so cells, and it becomes a blastula. Now, even here, we start seeing a difference in genes being turned on and turned off, and so we start having a difference in identification, but you don't really notice anything different in the cells. Now, some invertebrates and some vertebrates, you start seeing an uneven distribution of even the cells, and so in some of these organisms, you see much smaller cells on the top, much larger cells on the bottom, depending on how they divide. And it has to do with the insides of the cell being so thick, the cells divide unevenly. But what we're really interested in, how these cells become different parts of the embryo's body plan, and that really starts happening in gastrulation. And so you start seeing the development and differentiation of tissues. And we start by outlining 3 different layers called germ layers. Now, it's not germ like bacteria, but germ for germinate, because these are like arising tissues. Now, the ectoderm is going to give rise to the epidermis of the skin, the nervous system, which may seem a little counterintuitive at first because this is something on the outside, but it's making something that's underneath. But you'll see how that happens. It also develops the jaws and the teeth. The mesoderm, which is going to make the skeletal system, the muscles, circulation, and the lymphatic, also the excretory and reproductive systems. And then the endoderm, which as the name implies, this is all inner lining. And so this is going to be making the epithelial tissue, all of that inner coating of the digestive system, the respiratory system, all of that inner layer, that inner lining. And it's also going to be making the thyroid and the thymus gland, as well as the parathyroid. We see the adrenal glands made with the ectoderm and mesoderm, the different parts, but those are the main divisions. Now, we're going to be less interested in you knowing the memorization of these, but it's interesting to see how they get divided into the different parts. What we want to look at is how this is happening. Now, accompanying the differentiation is their placement. So we start seeing changes in cell motility, their shape, their adherence. And so over on the left here, we see the sea urchin. And the sea urchin, we see the gastrulation really well. We can study it and we can watch it happening very, very easily because they externally fertilize. And in gastrulation, we see the cells. You watch these arrows indicate the cells actually moving inward. This is the development of the different tissues, and not only that, but we start seeing that gastric tube being made. We see the hole forming as the cells invaginate, and then the tube being developed, and this is occurring because the cells can actually migrate around. Over on this right-hand side, we're actually seeing how the neural tube is created. And the neural tube, let me zoom in on this a little bit. The neural tube is made because the cells, we really see the changes in the shape here, because we start out cells that are relatively the same, but notice in these regions, you'd start to see the cells almost becoming a trapezoid or triangular shape. And as they do that, that will cause them to fold inward. Not the greatest picture, but you can perhaps see what's happening. And as they fold inward, we're going to create a tube, and that's becoming that nervous system, that spinal cord and tissue. And then these little areas, these cells are going to become the surrounding tissue as well. But either way, you're also seeing the three different layers, those germ layers, being created here. So after gastrulation, we see the organogenesis. Everything just goes full speed ahead. All of these cells becoming a part of a system and being dedicated to doing a job and having an identity. And so the development of the neural tube involves unfolding of the ectoderm, and that's why we can have something on the outside folding in to form something on the inside. And then you see the surrounding cells here, the somites they play an important role in developing the vertebrae and the surrounding tissue for that. So this is just a real brief glimpse into the beginning stages of the embryo. But what we really want to look at is how in the world is this happening? What's going on genetically? How do we know? And what can we do to kind of figure things out? But that's for the next video. Thank you very much.